This is Holy Cross Component 2 mock, and it is mock 2. Question 5. Read the passage about Saddam Hussein and DNA. Right. Use the information from the passage, and even more importantly, probably, your own knowledge to answer the questions. So importantly, when it says your own knowledge, so that means your notes. Use stuff from the notes. It's your invitation for you to cut and paste stuff from the notes if it's relevant. Describe how the technique of genetic fingerprint is carried out and how it can identify a person. So tell us all about genetic fingerprinting. So a reasonably straightforward question. So DNA is cut using restriction endonuclease. So now they've got fragments of DNA, and then they put this into gel electrophoresis to separate it. So electrophoresis, which separates the fragments according to size or length or mass. You could suggest DNA is negative, so we'll go to the positive end. I think that's what they say in physics, the positive end. I do think so. And you could say, small fragments. Travel further. Those two aren't on the mark scheme for this particular question, but they're the sorts of things you would need to know for EduClass now. And how do you actually detect these fragments? Well, we need some sort of a probe or dye or that sort of thing. So we are applying a probe. You might not have got that, but if you then said, detect using radioactive kind of markers or fluorescent markers, that sort of thing. And how does this actually, why are we going to get a difference? So the second is how it could be used to identify a person. It's not just enough to say everybody's DNA is different. That's not good enough at A level. We need to say why it's different. So different people will have different unique DNA as there are variable short tandem repeats. So short tandem repeats in the same gene. So that's why it's unique, because we might both have the same gene, but I might have more short tandem repeats, and so a longer piece of DNA which won't travel so far with electrophoresis. Part B, explain how DNA could be present on a toothbrush. Well, where is the DNA coming from? Is it the spit? Is it the saliva? Where's it coming from? Well, the toothbrush will have cells on it, so there will be cells. For example, cheek cells on the toothbrush. And you've got to look at how many marks are available. You go, well, that's not enough. Where's the DNA? What's the DNA got to do with cells? So cells will have DNA. So DNA present in the nucleus of cells. Or it could be mitochondrial DNA possibly. But that will do for our second mark there. Explain why the polymerase chain reaction, PCR, was used on the sample of DNA from the toothbrush. You should know all about that. So all you've got is this kind of toothbrush sample with a few cells on, and that's not enough to do any reasonable testing with. So what you need to do is do PCR, and that'll give you many more copies of the DNA. So problem is a toothbrush only gives a small sample of DNA. More is needed for analysis. And so what's PCR actually going to do? PCR will give many copies of the same DNA. So we're using our own knowledge of PCR. We're not just giving it a throwaway comment, need more DNA. We're using our own knowledge as suggested in the question. Explain one way in which the polymerase chain reaction differs from DNA replication in a cell. So with the machine, how is the machine different from DNA replication? 2 marks. So for example, probably the most obvious one is that it actually heats the DNA up to separate it, whereas you would use an enzyme helicase to separate DNA. So and we're doing this from the PCR point of view. So uses heat to separate the DNA strands. Now you could also add things like, they use a primer in PCR to initiate the replication, whereas primers aren't used. But there's a nice obvious one. Tests for use in criminal cases often take much longer. Explain why it takes longer if the sample is very small. Well, it's kind of repeating itself really. Need to do PCR. And actually this question is probably a little bit out of date, because PCR can actually be done pretty quickly these days in a very small machine. Contaminated. Well, contaminated with water, probably contaminated with other DNA. And then you've got 2 types of, or 3 types of DNA in the sample, and you need to separate them out, PCR them both, and then analyze them and take it from there. So that's other DNA is present, and you need to kind of find the DNA that you're most interested in. So you need to identify the DNA that we're interested in, the required DNA, the required DNA from the rest. So a lot of our own knowledge being put down there in relation to this novel example.